Good morning, Critical Thinking Classroom. How are you? Let me see where I'm live streaming here. I have a couple more things. I decided to get in the back here because the front was getting way too hot. The sun was just blaring, and and the guys needed to work over there. Although it is interesting work, and it's nice to see people working hard, as always. But I decided to come out here. I have to get something because I didn't come out here in the first place simply because the cushions were wet from our little bit of rain, or actually we had a beautiful rain, quiet rain, but heavy rain. The last almost, I don't even know how many days, which is why I'm wondering why I would have any brown spots in my lawn, but we're going to discuss that tomorrow in our early bird classroom when we take a closer look at science in our early bird classroom. On Wednesdays, we are doing science. On Thursdays, we're doing phonics in our early bird. And on Tuesdays in our early bird, we are reading. And right now we are reading If I Were a Cherokee. It's really interesting. It is so interesting. It makes you really begin to think about different ways of life that are really, don't just benefit the top 1%, right? Okay, let me get a few things done, like let me get a towel to sit on. And I don't have any of my books out here, but I did want to get online, somewhat quickly, or somewhat on time, because I've been running late. So let me get a few things organized. Like why I have an excellent signal, but it's not broadcasting, like that. I think it's because I'm in Chrome. All right, there we go. This should do it, I hope. Especially considering I've had an excellent signal the whole time. So, gremlins, everything's a big mystery, right? Okay, all right. I'm on. Let me go get that towel. I'll be right back. I'm losing all kinds of sequence on this dress. Sitting down, I looked up, I had all kinds of sequence that fell off this dress. So that's unfortunate. Oh, that's a nice picture of me getting those towels down. Oh! Tag up! Whoops! Oh, is that a bad shot? Yikes, is that a bad shot? Now I'm recording. Maybe I'll replace all that. I might replace all that. This is Dr. Annette Feravich. Thanks for joining me here in the classroom. I'm outside again. Now I'm in the back instead of the front. Woo! That Michigan sunshine. You would only believe it's not even 80 degrees out here. Yet we are just sweating in that sunshine. It is crazy, right? It's crazy. Yeah, no problem with the ozone. Cool. No, I believe you. All right, so still getting a few things ready, and I also want to get my coffee because it's delicious. Well, it's not as good as the one I had at Anna's house. I gotta say that coffee was really good. I am going to say it, and I did. All right, so let me open up a few of those slideshows first of all that we had open. So one of the things I want to tell you that I was learning about these, because remember I said I'm going to do some investigation about these twin studies, and it's not easy to find out what is really going on in the twin studies. And one of the reasons why it's not easy is because they are trying to hide the data, right? Because like I said, it was suspect where they got the original data from the twin studies. And here they're reporting it, and yet nobody's questioning it, right? We're not talking about the ethics of this, just simply just reporting the outcomes. It's fine to report the factual outcomes as the facts are, right? We could say African Americans, and the data showed this, while white people showed this, right? Or whatever you want to say, however you want to put it in the data. You're just, whatever the data says, you are simply describing the data, right? So that's what we want to do. But if my methodology is set up in a way that I know is going to bring about, which is what we teach, in 2,500 research methods, about how to set up your studies in a way that they are unbiased, right? Well, they know this. What are they? Did they go to college? Do they have an advanced degree? Are they conducting research? If so, then they have an obligation, and they not only an obligation, but they actually do have the knowledge on how to conduct ethical studies. And what they did by hiding that data and hiding those, the documentation, they waited to see how long it was going to take before people accepted and just took their data, right? And then they become, Philip Zimbardo, and they're known forever for their completely unethical studies, right? I don't know that Stanley Milgram falls into that, but I'm not really impressed with his desire for ethics, Milgram, so I don't know, right? So Stanford Prison Experiment, but there's also the, as we've talked about, the Robbers Cave study as well. So we talked a lot about that in our group think series that we had in our philosophical classroom, all right? Now, let's take a look at, there's a couple things I want to show you. Remember where we left off. Where we left off was this idea of interaction, which is a combination of nature and nurture, right? So, let's take a look at this. I'm really happy with the way some of this technology is working for me. How do I not have genes up here? Okay. So, or am I going to have this? So phenotypes, genotypes, and the most important factor, those are just some facts, right? Phenotype and genotype, those are the facts, right? But this is the important sort of conclusion, that genotype expression only occurs when there are environmental conditions, when there is an interaction that turns those genes on. All right, so we've been talking about this. I got the numbers mixed up. This is the day 5 of our twin studies. On the last slide, I said day 5. I don't think that was a correct number, but I am going to try and get those numbers up there correctly for you so that we could see how long we've been studying these twin studies for. All right? Okay, so, and then this was the interaction that we talked about, right? So we said that one genotype number 1 or number 2, I'm not sure which one it is. One of them is going to be no parents, right? So we get our genes from our parents. No gene problems from our mom or our dad. No genetic predisposition to depression from our mom or dad on genotype 1, let's say. Genotype 2 would be that one parent has predisposed us to depression, but not both parents. And genotype 3 is where both parents do. You can see that the only people who ended up with high levels of depression were those who had both a genotype that predisposed them to depression because they got it from both parents, as well as high rejection, so an environmental condition. So this is where we are getting our, and it says phenotype, so our gene expression. That's our gene expression. So we get it from a genotype, but we get it from in the environment. We also get it from the gene environment correlation and gene environment interaction. Now, we really need to see I'm not going to worry about the differences there. All right, we're not going to worry about the differences. There's a technical difference there, and the difference is so small. One of them is an association, and the other one is literally if-then, right? So one of them shows a relationship between gene-environment relationship. That's a gene-environment correlation. Well, the other one is showing what this is showing, that if this happens, if you have genotype number 3, where you have depressive predisposition from both parents, and a high rejection, then, right? If this and that is an interaction, that's what an interaction is. If this and that, then this. In only one of the 4 conditions here. In only one of the 4 actually there's 2, 4, 6 conditions here. 6 conditions. All right? Let's take a look at a few other things to help us see what this environment does. This is one of my favorite slides. Oh, actually no. Yeah, I want to tell you, I'll give you a couple examples of, so these are not the rats that we're talking about, but I'm going to give you another rat scenario that shows you the environment and how the environment works and how experiments work as well. So we randomly assign participants to certain groups. So we've got a group that has got bright rats and dull rats, all right? Bright rats and dull rats. And what we're going to do is we are going to randomly assign you, I don't know who, here you go, you get rat number 3. Rat number 3 is a dull rat. You get rat number 7. Rat number 7 is a bright rat. You get rat number 10. Rat number 10 is a bright rat, all right? So we randomly assign, and you may have a bright rat or a dull rat is what they called them. And then they put the rats in the maze, and they were told that they had to teach the rat how to get through the maze. And amazingly enough, the most successful rats were the bright rats, of course, except for the fact that there was no difference in intelligence level. The only difference was that individuals were told they had a bright rat or a dull rat, and that was the only difference. And those bright rats succeeded in the maze more than the dull rats did, even though they just took a bunch of rats and they said, here's a rat, it's a dull rat. They just told you that. They just lied and said it was a dull rat or a bright rat. But the success was based on what they were told. This is called a self-fulfilling prophecy. You're told something and it happens. It occurs. Scientists are trying to do this all over. You're told you're an alcoholic, and so you are. You're told, and maybe you are, right? Maybe you are. But it's not simply in a self-fulfilling prophecy, right? I had a lot of people who, when I was, familiar with the program, who wanted me to be an alcoholic. They want you to be an alcoholic sometimes, to be like them, right? So they keep doing this sort of Self-fulfilling prophecy. Think about it. Well, nobody thinks about alcohol that much. You must be an alcoholic. You just told me to think about it. Creating these narratives for self-fulfilling prophecies. This is the problem that I've had with psychology, is creating these narratives in order to get certain outcomes that they've been looking for here in 2023. And for the last, 50 years, I would say, just producing outcomes that they want to see. And of course, that all goes back to money as well. That all goes back to profit as well, right? Okay, so we can create self-fulfilling prophecies because of our beliefs, because of our environment. This here is one of the most interesting slides that I picked up from an article that should be in our literature that we study all the time. Take a look at these. What we're looking at here, and I have the most interesting ones highlighted because there's a lot of information here. So what we see is the organism, all right, the live creature, right, the organism. Here's the creature. Here's what we manipulated in the environment at very, very young ages, sometimes in utero, sometimes at very young ages. So sometimes just right in the womb. We'll read those in just a minute. And then here are the outcomes. So here's our creature. Here's the manipulation of the environment, and here's the outcome. So let's take a look at that a little bit more closely. So nematodes, infant cells, right? Or in utero cells, nematodes. Oh no, what am I talking about? Nematodes is a species. What am I talking about? I don't even know what a nematode is. So look at the environmental changes that they introduced for each of those. Nematodes, whatever those are, probably in utero toads, right? Tadpoles in utero, probably. The absence or presence of food. In fruit flies, they elevated heat stress. If you elevate heat, you get stress during larval development. So while their larvae is developing, they elevated heat, stressing fruit fly larvae. They changed for fruit flies light and dark cycles. So instead of nighttime, right? So during the day, they would have night, and during the nighttime, they would have daylight. So changing light and dark cycles for fruit flies. For various reptiles, they changed the incubation temperature like they did with the fruit flies. That one I highlighted. Look at what changed. When they turned up the heat, sex determination changed. Do you understand that? With environmental changes, they were able to study and change the sex of various reptiles by changing incubation temperature. Songbirds like canaries and zebra finches, they change their conspecific song. What a conspecific song is what in nature they listen to, right? So canaries have a certain song that's different than finches do. What did they do? They changed the bird song that's introduced in the environment in utero, and what it changed was it increased their forebrain and increased their mRNA, which you realize they are disrupting, right, with the virus. So they're changing. Darn it, come on. Okay, so did you hear that? What I said? Oh, I needed to see that a little bit larger, so that's what I did. And so do you. So here, in the songbird canaries, when they changed their conspecific song, what ended up happening was that they had an increased forebrain and increased changes in their mRNA. So changes in the mRNA are changing the size of our forebrain. What's our forebrain? Our forebrain is the area of our brain that does all the thinking and problem solving. So thinking and problem solving, DNA, mRNA. So the translation of DNA into mRNA and relationships with the forebrain, right? So we see a correlation with the forebrain and changing the song. That's a genetic environmental correlation. There's a correlation with the forebrain, but it changes the mRNA. That's an interaction, a genetic environmental interaction. Okay? Let's take a look at some of these other ones. So, in hamsters, they changed the light and dark cycle. And the outcome was increased pituitary hormones. Our pituitary gland is the master gland that regulates our hormones. And so here, by changing light and dark cycles, we were disrupting hormones, we were disrupting mRNA, and we were disrupting reproductive behaviors. It makes sense? The pituitary gland is the master gland for our hormones. Of course, our reproductive behavior would be changed from what? From simply changing light and dark cycles in our environment. Oh my gosh, I didn't realize that you can't see what I'm looking at. So, up at the top there, I've got that yellow there that says light and dark cycle. So the one isn't highlighted, and then I've got highlighted there incubation. So these are for various reptiles, songbirds, and hamsters, right? So various reptiles, incubation temperature, songbirds, conspecific songs. They changed their songs that were known to that bird, right, to their offspring. So they were singing a different song to the bird's offspring. And then light and dark cycles for hamsters. And then here we see sex determination was changed. Increase in forebrain and mRNA for the conspecific songs for the songbirds. And increased pituitary hormone changes in mRNA and changes in our reproductive behavior. This is just too incredible to dismiss. Mice, they changed acoustic stimulation. Mice, they changed light and dark cycles. Rats, they changed tactile stimulation in rats. What does that mean? They changed how they touched, right? Tactile stimulation. More or less touch in rats. What was the outcome when they changed their tactile stimulation? Enhanced EFOS expression and increased number of somatosensory sense of touch cortical neurons. So we have more neurons in our brain that pick up and are more sensitive now to touch when they changed how much we touched an infant. We also found out from previous studies that we looked at, Harlow's studies, that when monkeys weren't touched, and we know this from Romanian orphanage, that when children weren't touched, that they had stunted growth, right? Their growth was stunted. So we know that there are a lot of changes that go on when individuals do not have tactile stimulation. But the somatosensory area of the brain is an important area of the brain for what we would say actually empathy but for mirror neurons. So when they're talking about changing the somatosensory area of your brain, they are talking about changing the area of your brain that's responsible for liking touch from another human being, right? Not being repulsed from it, finding it nurturing, the touch, finding that nurturing, but that allows us to empathize and have compassion for other individuals. Let's keep going. Rats, they're in highlighted, they changed their environmental complexity. So did they have wheels? Did they have, were they able to travel up and down? Now here, some of these were when the species, right, the critters were in utero. So they changed the infant's environment, the rat baby, in the womb of the mother rat. Sometimes they changed in the environment. Like here, did they have nothing? Were they in a cage with nothing? Or did they have wheels? And did they have climbing mechanisms? And did they have other rats or things of other interest? So environmental complexity increased brain RNA diversity. If we have increased diversity in our brain, this also allows for more mirror neurons, more compassion. These are the things that need to be studied, but these are the things that are related to these environmental changes. Prenatal nutrition was changed in rats, and what did that do? It increased or decreased, it changed the number of brain cells. Increased cerebral DNA, increased the number of brain cells because of nutrition. So breakfast is an important meal. Cats, we've already talked about this several times, changed their visual stimulation and increased visual cortex in their RNA, which meant that they were able to see more complex elements, as opposed to no color, no angles, no straight lines. So by changing their visual environment, they change their ability to see. When you see, they change their visual cortex to create more complexity or more diversity or less. What does that mean? It means that I have more or less ability to see. Literally, it changes my vision. The last one, humans, they changed academic examinations taken by medical students to create psychological distress. And what did they find out? They found out that they had lower immune system responses. Okay, so this is an unbelievable study that they have done on changing the environment and how that literally changes our brains. We know that culture also changes our brain so that we see more or less of, for example, the scene. Is your attention completely focused on me, the speaker? If you were here born in the United States, likely it would be. We pay attention to the people, to the individual. In Eastern cultures, their brain is wired after experience, after having different environments. Their brain is, in general, not for everybody, wired to see the big picture. So they would see the boat back there, and they would see the trees back there, and they might wonder what this little contraption is right here, which I've always wondered what that is as well, right? So they might wonder, right? So they see the whole, and they remember the big picture, where here in Western countries, we have a tendency to remember individuals and pieces that are most important to us, and the details of those pieces that are most important to us. But just pieces, not the details of the big picture. Eastern people, they miss the details often, but they got the big picture, right? So very interesting the way our brain can change our understanding of all kinds of things, right? Let's see what the next slide brings. This is a way we see double the difference there, which basically is just how do we calculate how much of any trait is inherited and how much of that trait, so how much of it is due to our genes and how much of that trait is due to the environment. So this is relatively important. Okay, let me show you something else. I'm gonna stay on this, and I'm gonna show you these stats here. So we see that height, for example, has a 0.86 correlation with our DNA. It is highly correlated, as DNA suggests. Our height is going to be, whatever it is, 5'5" for me. My DNA may have suggested that my height was going to be 5.6, but maybe I didn't have enough nutrition as an infant. I have a feeling my younger sister did not have enough nutrition, although she's taller than me. So nutrition can affect height. So there's not a perfect correlation with DNA for height and actual height. Do you understand what this is saying now? So what this is saying is we can measure your DNA for any trait, any one of those traits. So what else do we have? We can measure your DNA for weight, for example. We can measure your DNA for brain waves, is what that says there. We can measure your DNA for, I actually can't, I'm going to blow that up like I do normally. There we go. Blood pressure, IQ score, personality traits, occupational interest, your interest in what you want to be. Did you know that your career interests have a genetic basis to it? How do we know that? How do we figure that out? And this is where the twin studies come in. So we can see religiosity, for example, which is a personality trait, right? That is relatively the same as any other personality trait, but it is in part inherited. How do we know that? How do we know? Or non-religious social attitudes. So religious social attitudes, look at that. Do you see what I'm seeing? That religious social attitudes, when you're religious, that has a higher DNA component or contribution than non-religious individuals. How could that be? How could that be? See, that's the reason why, because I'm always reading something. I'm always reading something behind. And so when I'm reading something, I am squinting a lot. Just there is a lot of squinting. I gotta find Lucy for a sec. So take a look at that and see what you come up with. And remember, we're saying that in height, DNA contributes more, but DNA isn't 100% of the reason. It's 85% of the reason. You don't really use that number that way, but you can think of it that way. DNA is 85% of the reason why we have our height, but there is still an environmental factor. Our weight has a high DNA contribution, but environment also contributes to our weight. How about brain activity? Brain activity, high DNA contribution. You can think about this as, like, in particular, are you thinking? Are you problem solving? Are you decision making? That's about 80% of the time. 80% of our alpha activity in the brain is due to DNA. But 20% is not. That's due to our environment. So even something as hardwired as height still can vary because of the environment. Now, this is good news for us. This is good news for us because even the most hardwired traits can be changed with environment. So you could say, well, so you're saying, Dr. Feravich, that I could be taller with the environment? Well, you could have been. It just depends on your DNA, right? It depends on your DNA. But there are limits, right? Your DNA limit may have been, my DNA limit may have been 5'7", and maybe but I am 5'5", right? So could I have it? So the question is, could I have been taller? It's possible, but you can't be taller than what your DNA suggests. Okay? Let's take a look at another report that we have here. There we go. I think this is the one that I want. Yeah, it is. Okay. Let me make sure I've got the setup, which I should. No, I don't. Probably because I didn't save it. And now... There we go. Okay, so this is actually chapter 2 from my child development book, which is from, oh gosh. Cengage Publishing, okay? Siegel it's 2 females, Siegel. I'll get the book for you, and then you'll know what book I'm using, if I get the book for you. Let me show you this. So when I was showing you on Thursday the splitting of cells, I was showing you that we had chromosomes, which likely you know, you've seen chromosomes before. Chromosomes are made up of 2 chromatids. These are chromatids, which is a single strand of our DNA information, okay? So when we take a look at this picture here, we've got the genotype information on our chromosome, which is made up from one parent and the other. So one parent gives us a set of genetic information on a chromatid, and the other, and it could be on either one of the chromatids. It doesn't matter, right? I'm getting genetic information from one parent. It could be on one chromatid or the other chromatid. And then the other parent is on the other chromatid, all right? But together, we need them both together to make a gene, right? To get the genetic information, we need both chromatids from mom and a chromatid from dad. And what we end up with are 23 pairs of chromosomes. 22 are autosomes and 1 are our sex gametes, all right? I don't think I put our sex gametes here, did I? Oh, I did. There we go. And there's number 23, our sex gametes. All right, so what does that mean? You can see here that our DNA, our chromosomes, everyone has got 20, unless there is some genetic defect, everyone has got 23 pairs, 22 autosomes, which carry all of our genetic information, and that genetic information splits and splits and splits in this way. All right? So chromatids, they duplicate, become chromosomes. The chromosomes split so that you have the same genetic information in each one of the cells, and they split the chromatids, for each one of the cells, and you're back to 2 cells with chromatids that then duplicate and get our chromosomes. All right? Now, that's not too much and when we talk about genetic information for our sex gametes, that's even more complicated. It's just it's actually a double step is what it is. Chromatids duplicate into chromosomes split, and then they split again. And that's what happens to our sex chromosomes. Let's take a look at those here, though. So these are the 22 autosomes that carry all kinds of genetic information like we just saw, right? Like our weight, our height, our eye color, alpha waves for our brain, personality. All of that is contained in information that we've seen, right? All that genetic information, not simply genetic information that is simply located in one spot on our chromosomes or on a chromatid, but can be a combination of information from all over the place. We're going to talk about some true and false about passing on some information. We already did look at those true and false. I don't know. Did we go through all of them? I'm not sure if we went through all of them. We've got to look at those true and false questions and see if we did. I don't think they're here on this one. But we'll get to that. So we see a little line there, which is indicating genetic information. But for something like intelligence, we don't have just one gene, as we said in our true and false, right? So we looked at those true and false. I'll look to see if we finished all those true and false. We did, because that was one of the last questions. A trait like intelligence, does it have one gene? No, it's not located in only one place. So we get, so for personality, we go and take information from a lot of those different genes, from those autosomes. Then we've got our X and Y chromosomes from the male and the female, which are our sex gametes, our sex chromosomes. Look at the difference here in size. They did a close-up on that. So a couple things you can notice about these. First of all, when they pair up, when the chromosomes pair up, they pair up with a like chromosome. They just don't pair up with any chromosome. They pair up with the chromosome that contains that important genetic information, right? With their like chromosome. So the male and female are together here, and they pair up the same chromosome. Chromosome number 1, chromosome number 2, chromosomes number 3, all right? All the way down to number 23, which is our sex gametes. Look at how much bigger that these are under a microscope, and they are relative size. Look at how much bigger the X chromosome is than the Y chromosome. This is the reason why males may have more genetic defects than females do. They don't have room for all the genetic information like the X chromosome does. And so they may be missing important genetic information that simply didn't fit on the Y chromosome. How about that? How interesting is that, right? Now let me show you. So we said autosomes are non-sex chromosomes. Gametes or germ cells are the sex chromosomes. They split through mitosis. Autosomes split through mitosis. Sex chromosomes split through a process called meiosis. There's mitosis, there's meiosis, all right? And what we end up with is 3 non-functioning cells of information. What? And 1 functioning one. Interesting. What happens to all those non-functioning cells? And there in the end, there we get our zygotes, where we've got genetic information from both the mother and from the father. So I want to put this up. This is what DNA looks like. And what DNA is it is the coding for our protein. Why do we need codes for our protein? Coding for our protein gives us our information. To make an ear, this is made out of protein. To make a finger, it's made out of protein. To make my liver, it's made out of protein. What is DNA? It's deoxyribonucleic acid. I had to look at it. Deoxyribonucleic acid. I haven't said that in a while. Deoxyribonucleic acid is DNA. DNA is made up of 4 different proteins. Adenine, thymine, right here, they're listed. Cytosine, and guanine. Or guanine. I don't know if I said that. Guanine. So, only 4 proteins make up every piece of information that you have in your body. And it's all located on these 23 pairs of chromosomes. All your information is located on these 23 pairs of chromosomes, which are all located in the zygote, in the very first cell. All that information is located in our very first zygote. Our very first single-celled organism contains all those 23 chromosomes, information from your mom and information from your dad. What happens is we combine those to get like this alphabet soup over here is what we get. So A combines with T in different ways, and C combines with G in different ways, so that you've got all of your genetic information, if you can believe that, out of 4 different proteins. A specific portion of DNA on a chromosome is called a gene. That's what we were seeing in that picture there of those chromosomes that were colored, right? We were seeing genes. Those bands there are indicating different genes on a chromosome. So what is a chromosome made up of? Well, there are bands of genes on that chromosome, which is a specific portion of the DNA on the chromosome that gives us our genetic information. This information is triggered by the production of proteins, by the production of our DNA, by splitting and resplitting, right? So that's how, right? So as the cells produce again and again and again, we'll get more and more and more skin. As the cells reproduce again and again, we'll get more and more thinking power, right? So this is what happens. Why do the cells duplicate? So we get bigger. That's why. So we get bigger. So we have physical growth, both physical growth that we can see and physical growth that we don't see. Here we can see that we've got different chromosomes that pair together, one from a parent, one from one parent, and one from the other parent. Sometimes those chromosomes are capital letters, right, representing a certain chromosome. Sometimes they're small letters, and sometimes they're a combination of a capital and a small letter. Capital means it's a dominant gene, is all that means, right? So they're describing with a capital letter a gene that is a dominant gene. When we have a small letter, what that means is there's a gene that's a recessive gene, Let's take a look at that a little bit more closely, all right? So we can have a dominant gene from one parent and a dominant gene from another parent, right? So, for example, we can have a dominant brown eye color from mom, and we can have a dominant brown eye color from dad, and that will tell us that we are going to have brown eyes. Here we've got an example of curly hair, okay? But sometimes we end up getting traits from a parent that are not dominant. And what that means is that the dominant trait will be shown. I think I've got... No, I don't. It's not here. Which dominant traits will be shown? I'm surprised, though. Let me just see something here. There we go. There we go. All right, this is what I'm going to stop with, though. So what this is showing is which dominant trait, which traits are dominant and which ones are recessive. Dominant traits will be expressed, recessive ones will not be, right? So brown eyes is the dominant trait. If a parent gives you a brown eye gene, your eyes will be brown. No matter if another parent gives you a gray eye gene, a green eye gene, a hazel eye gene, or a blue eye gene. The dominant trait is brown eyes. Dark hair is a dominant trait to blonde hair. This may be the reason why people admire or desire blondes. Blondes. Why? Because it's not that often that you see a true blonde. Why? Because dark hair is a dominant trait. If a parent has dark hair and gives you a dark hair gene and another parent gives you a blonde hair gene, your hair is going to be dark. Non-red hair is the dominant gene compared to red hair. Let me actually get this smaller here. I don't understand why I grab it and then 60 other things are grabbed instead. It's highlighted, the other one's not highlighted. Why does it need to be grabbed? I don't know. Curly hair is dominant compared to straight hair. Normal vision is dominant compared to nearsightedness. Farsightedness is dominant compared to normal vision. More people are farsighted. That is that they can see far, right? That is dominant compared to normal vision. Broad lips are dominant compared to thin lips. Double jointed is dominant compared to normal joints. Pigmented skin, to have color in your skin, is dominant compared to albinism. Type A, blood type, is dominant compared to O, and B type blood type is dominant compared to O. And blood type is a way that we can kind of see, right, patterns of these different DNA combinations. Right? So, for example, some people have blood type A. Some people have blood type B. And some people have AB blood type. Right? Some people have O. Some people have AO blood type. So they would have blood type A. If I have an O blood type, O negative, that's a rare blood type, right? But we can see in how we name our blood types. A means a dominant from A, and some could be any other recessive. So B, dominant B, any other recessive. But if I have AB blood type, I know I got A from one parent and B from the other parent. This is Dr. Annette Feravich. We will continue understanding more and more about how we pass on our genes. How do we get to look like we look? And most of that is studied and understood through our twin studies. Thanks for joining me on this classroom. This is our critical thinking classroom. I am Dr. Annette Feravich, and I will see you here in about an hour for our spiritual connection classroom.